Good morning, good afternoon, and good evening, everyone. And thanks for joining us today. This is Liam Sanyo from Inside Scientific, and I'll be your host for today's event, which is titled Simultaneous Multi-Region Neuronal Calcium Imaging in Freely Behaving Subjects. This webinar has been sponsored by Neurescent, so a big thanks to them for making it possible. Today, we're being joined by Dr. Yasaman Soudagar and Dr. Roshni Christo from Neurescent, who will present a method for simultaneous calcium imaging in up to 4 brain areas in freely behaving mice and discuss some exciting potential future applications. And without any further ado, I'm very pleased to welcome the first presenter, Dr. Yasaman Soudagar. Yasaman, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you so much, Liam and Inside Scientific, for making this happen. So as Liam said, I'm Yasmin, and my training is actually in physics, and I'm here with my colleague Roshni, whose training is in neuroscience. And we are going to talk about multi-region calcium imaging in freely behaving subjects. We are actually going to start this presentation by a poll because we want to ask you if you have ever thought of what type of research questions you can answer if you have the capability of doing calcium imaging simultaneously in multiple regions of the brain and the spinal cord of freely behaving subjects. And this is any brain region that you wanted to. So, well, this is great. So the polls are coming in, and it is really awesome because it seems that more than 90% of you have thought about this. So this is great. We have also gone online and done a literature search to see what type of research is being done that encompasses multiple brain areas. And we actually realized that the application is basically quite vast. It covers anything from understanding obesity or eating disorders all the way to motor disorders, Parkinson's, and memory studies, Alzheimer's, epilepsy. So there is really a large range of applications and large range of questions that can be answered if one can simultaneously study multiple brain areas. Now, imagine if you can do this type of study where you can simultaneously follow the activity of subtypes of neurons in these brain areas longitudinally while looking at the subject's naturalistic behavior. So that is basically what we will be talking about today. But how would one do that? Well, this whole area is really thanks to all our colleagues who have developed these wonderful tags. So they are in different groups like GFPs, RFPs, and the tags, these are basic fluorescent tags, and they can come in various formats. Some are passive, so they just express fluorescence, but some are active. Like an example is GCAMP, where it is a protein that when it binds to calcium ions, it falls and becomes more fluorescent. So the way these type of active tags like GCAMP work is that they get expressed in various subtypes of neurons of interest. And as is shown by the group that have developed it, Chen et al., if you have a neuron, so there is a neuron where it's expressing GCAMP and it is being patch clamped at the same time. The little red you see there is a patch clamp. And they showed that the fluorescence signal, which is the blue line from GCAMP6 fast, is showing a rise whenever you have an action potential, which is what is shown on the lower line. So the blue line is the fluorescence signal, and the lower line is the signal from the patch clamp. So, so this is great because what this means is when you use a tag like GCAMP and when you do fluorescence imaging, when you see a rise in the fluorescence signal, it is essentially showing you the activity of the neurons. Now, there are multiple methods of following or studying neuronal activity in behaving subjects. The most used and the most established method, which is also quite powerful, is in vivo electrophysiology. Electrophysiology records the electric signal from a volume of the tissue, and the subject can be moving around when that is happening. The signal you record is really the average signal, But then there are mathematical methods that allow, to some extent, to divide this signal between a certain number of neurons, or sometimes even tell the subtype of neurons. The other method is called photometry. Photometry makes use of these fluorescence tags such as GCAM. And what happens is once the users express GCAM, they implant fiber optics and they monitor the fluorescence from the tissue. Now, this fluorescence that is being monitored again is the average fluorescence signal from a volume of the tissue. The difference between photometry and electrophysiology is in two things. So the first thing is that in photometry, because the user can tag subpopulations of neurons with a fluorescent tag, the signal that you're collecting, you know it's coming from this particular subpopulation of neurons. But the second difference is that electrophysiology is much faster. I mean, the 4-bit half maximum of an action potential is only 4 milliseconds, while when you're collecting fluorescence signal, the signal is much larger. So usually it's 150 to 200 micron 4-bit half max. Now, the other set of techniques are imaging techniques. The imaging techniques are, again, in two sets. One is 2-photon imaging and one is epifluorescence imaging. A main difference between 2-photon and epifluorescence is in the resolution. In fact, 2-photon imaging was invented to have very high resolution. The resolution is so high that you can study dendritic spines with this technique. And in fact, it is really good for research questions that require such high resolution. Because of this resolution, the technique becomes very sensitive to motion artifacts. So the animal's head has to be fixed under the microscope, and the field of view is about 350 micron by 350 micron usually. And the whole system takes, it is large, it takes a whole room. And to operate, basically, it requires a lot of expertise, both in physics and in neuroscience. But then again, if you're studying something like dendritic spines, this is the technique that you would want to use. Now, as opposed to that, there is the epifluorescence microscopy, which is just usual regular fluorescence. Now, with that one, the resolution is good enough to show you the cell bodies. So because of that, it is less sensitive to motion. And because of that, you can basically implant lenses anywhere in the brain, so you can image either shallow or deep areas of the brain in freely behaving subjects, or in subjects that are doing their naturalistic behavior. Because you can visualize cell bodies, this technique is great when you're studying brain circuitries. And the field of view, depending on which brain region you are at, it can be about 1 millimeter in diameter, or smaller, or sometimes larger. Now, in this presentation, epifluorescence microscopy will be the technique that we will focus on. But again, before we continue with that, we would like to know what sort of in vivo recording techniques you are currently using in your lab. So we have another poll, and let's see what you guys are using. So the majority is using electrophysiology, and then photometry comes next. Epifluorescence imaging is third, and 2-photon imaging is fourth. So this is wonderful. So this means that most of the audience are in fact familiar with recording from moving or behaving animals. So let me move on to show you, again, we are focusing on epifluorescence imaging, and let me show you basically the different options. There are the mini scopes that you see on the left. The mini scopes, to use them, you implant a small lens in the brain of the animal, and then the whole of the microscope gets connected to the head of the animal over that lens, and the imaging is done. So here you see that in those pictures, these are all mice, and you see the size of the whole microscope relative to the body of the mouse. So mini scopes image one area, and the whole device with all its electronics sits on the head of the animal. On the right side, there is the neurosens multiscope, where in the animal on the top, the mouse on the top, there are two implants in its head. So the lenses are very similar to the miniscopes. These are small lenses, they get implanted. But the difference is that the device itself, the microscope itself, is away from the head of the animal. Instead, what is being used are these special fiber optics, which connect to the implanted lenses, and the imaging is done that way. As you can see in that picture, you see the two fiber optics leaving the head of the animal, and they are quite flexible and lightweight. In fact, everything is so small and flexible that you can implant 4 of these on the head of the mouse. And you can implant them in the spinal cord, as you can see in the middle picture. Now, of course, there are other species other than mouse. So one of the collaborating labs is in fact using songbirds, and they have been able to implant 2 of our implants on the head of the songbird, as you see there. And the songbirds, of course, they could fly with the fibers connected to their implants. They could fly in the cage, and they sing, which is obviously extremely important when you are doing research with songbirds. So we are really excited about that. Now, the instrumentation itself, it's called the quartet. You see the 4 fibers coming out. So the device itself is sitting at the center, and everything around it is the control electronics. So the main body of the system itself is shown here. You can compare the size with my hand. So it is small, it is lightweight, and it connects to the computer through GE communication and another wire, which essentially controls the level of the illumination light. The camera used in there is a color camera, which allows you to not only see green from GCam, but the filter we use allows red to also get through. So in case you have tagged neurons with something like chili tomato, you might be able to see it. The product or the system is optimized for GCam imaging. These are the imaging fiber bundles that I'm showing you here. As you can see, they are really flexible. They are 84 centimeters long, each of them. And inside each, there is 18,000 individual fibers. Now, the fibers are flexible enough that they can twist and turn without any problem when the subject moves. They connect to this lens connector, which the lens part that I'm showing you right now is the part that goes inside the brain, and the rest of that metal connector is fixed over the head of the animal. Now, when you are not imaging, that is the part that sits on the head of the animal. So this is one of them, and they are small enough that you can put 4 again on the head of a mouse. Now, when you want to image, you need to connect the imaging fibers to this lens connector, and that can be done very easily, as I show here. So you connect to do the imaging, and the disconnect is as simple to do. But how is this whole technique done? What is the workflow? You first need to have animals that express GCam, and that is done in 2 ways. Either you use transgenic mice, for example, that are already expressing GCam, or you use specially constructed viruses that express GCam. Now, the viruses are really useful because with that, you can literally choose which types of neurons you want to express GCam in. So you can choose subpopulations of neurons. But sometimes when you use viruses, you also need to use, depending on the type of neurons you want to tag, you might need to use some transgenic mouse lines, such as cream mice. If you do decide that you want to do viral injection, well, you need to inject the virus in the brain regions of interest, and then you need to wait for about 2 to 3 weeks for the virus to express. After the expression is done, then you need to implant these small lenses that are called green lenses for greater refractive index. So the name of these lenses is green lenses. As we show here in the middle, here we are implanting in bilateral hippocampus, so the craniotomy is done for that region. So you remove the skull, you need to aspirate tissue, and then you lower the lens down. Then you fix the lens connector you basically cement them to the skull. So that is shown on the image on the right. And another thing we're showing here, if you can see it, is there is this surgical accessory we have developed, which is called the piccolo, which helps with precise targeting of the brain region that you want to go to. So it nicely holds the lens connector, and it allows you to do precise targeting. Now, after you implant the lenses, you need to again wait for about 3 weeks for the brain to recover from the surgery. And that is when you do your test image recording. So you basically bring the device, you connect, and you image. And that is because you want to see basically how did the surgery go, how many neurons you're seeing in each region, is it successful, and you want to focus and find the best focus and image neurons. And once you have that, you are now ready for your behavioral testing. Now, an example of how the whole system is used in a behavioral testing is shown here. So here we have an elevated maze, and you see the behavior camera on the top, and the quartet system is pretty much sitting beside the behavior camera. It's hanging there. And the whole thing is set on a sort of a tripod, which is it is basically like a microphone stand, and it allows you to easily change the height. And you want to be able to have something like that because, again, the fiber optics that is coming all the way from the quartet system down to the head of the animal, it is 84 centimeters. So when you set this up, you want the fibers to easily reach the extreme ends of your behavior setup, whatever that happens to be. And you want it to be in such a way that when the animal goes to the center, the fibers don't sag under the animal, so the animal doesn't get tangled in it. Now, once you set this up like this, everything is prepared to go. Now, in terms of imaging, here on the left, I'm showing you an image of cortical neurons in a brain slice. So, I mean, obviously, brain slices are a very clean system, and there you see some really nice cell bodies and also the axons. When you go to the in vivo brain, we usually see the cell bodies, and most of the times we don't really see axons. But then again, we want to do brain circuit studies, so it is really the cell bodies that we are interested in. Now, what is being shown on the right is this capability that is specific to the neurescence multiscopes. And this capability is a focus lock mechanism. What it is that once you connect the fiber to the implanted lens, depending on where the neurons are relative to the lens, you need to focus on the neurons. So you do the focusing, and so here I'm doing the focusing on the brain slice. And when you are done with your imaging session, you disconnect the fibers, and you put the animal back to home cage. When you're doing longitudinal imaging, the day after or for the next imaging session, you bring the animals out and you connect the fiber back. Now, we have, this is designed so that the focus stays on the head of the animal. So you don't need to refocus anymore. And you can basically image multiple days or even weeks without having to refocus the system. And that is important because for some of the experiments, for example, for memory experiments, really as days go by, the neurons that are being recruited to the circuitry, they change. And you want to be sure that when you see a change, it is because of the brain changes, and it's not because you are now focusing on a different set of neurons without realizing it. So this focus lock mechanism ensures the accuracy of the data that way, and it ensures that you are following the same neurons longitudinally over the span of days. Now, in terms of the software, you can, this is the software that comes with the device. You can read in the 4 areas. So here you see all 4 imaging fibers. what happens is that you can turn the illumination light on and off. You can change the power of the illumination light, and what you can do is you can save. When you're recording, you can save at 20 frames per second, and when you save these, it is designed so that it is saved as frames. So it gives you the date and the time, and you can add anything else after that, of course. And what is saved are frames where you have the frame number, the time that the frame got saved. And the format is in TIFF because, as scientists, we want to be able to analyze our images and data with anything we want. So, and TIFF being the standard, that is what it is using. Now, let's say that, you haven't implanted all 4 areas and you have implanted, say, 1 area. And now what you can do is you can basically go in and crop out that 1 area that you have implanted. So in this case, it is the one on the top right that we want to crop out. And, you just easily now end up with 1 area. And when you save the movies, this 1 area gets saved. Hence, you don't have to have a lot of, pixels in your image that you don't want to use. So you basically end up using only the part of the image that is relevant. And, of course, then you can undo the crop and go back to the initial setting. Now, the question is, what if, you have saved a movie and you want to analyze it? So here we are reading in a movie from the subiculum. And you see the neurons, they are turning on and off, if you will. And what you can do in the software is there is this bandpass filtering. So right now I filtered out the big fluctuations, and I increased the contrast, and now I'm playing with the brightness. So it's quite easy to process, these movies. And now that we clearly see the neurons, we can go in and we can contour the neurons. And as we contour them, the activity gets extracted, as you see on the top right there. So now the thing is, sometimes, the neurons can be fast, so you can pause the movie and, calmly and precisely contour a neuron, and then you press play again and it runs again, and it shows you other frames where other neurons are active. Now, what happens if you realize that, oh, this really wasn't a neuron and it was a mistake? You can just go and deselect that contour. Then you basically say save trace, and what happens is 3 files get saved. So there is a comma separated value file that is giving you the regions of interest that you have picked. It tells you the time, the frame number, and the intensity of light in that ROI. So here I'm basically just plotting the intensity, which is essentially the activity of the neuron, which is that ROI. Another file that gets saved is a PNG file that quickly shows you the contours and the numbers associated with them, and it is color-coded. It is basically just a quick visual. And finally, there is another CVS file where, CSV file where you see the ROIs and you see the contours. Now, imagine that this is now another imaging session from the same animal, and you want to follow the same neurons over multiple imaging sessions. So you can go in and read back the contours you saved from the previous session. And let's say that here we realize, oh, there is this neuron and, oh, this other neuron that weren't firing in the previous session. So we contour those, and that neuron that was firing previously is not firing anymore, so we deselected that one. So you do that and you save this again. And now you have 2 different files, 2 different contours for 2 different days. And basically when you go in, you can see which neurons or which contours remained over the span of a few days, and which contours you have eliminated and which ones you have added. So this way you can keep track of the same neurons over the span of days, and you can, if you need to, if the circuitry is changing, you can add and subtract neurons to your file as days go by. But each time the file is saved, so you don't lose track of the neurons. So I have shown you everything I wanted to show in this presentation for the software, but there is a lot that we could discuss, and we would be happy to later. So at this point, I want to basically, we want to get down to business now. I want to know image behavior. So we asked you this poll question, and I would like to hand this presentation over to my colleague Roshni, who is a neuroscientist. Take it away, Roshni.
Thanks a lot, Yasmin. And yes, I'm quickly looking at the poll results here. As we can see, there's a lot of learning and memory studies, a few motor behavior studies, anxiety and depression studies. Oh, wow. Okay. I think learning and memory tops the list here. So let's get on with the presentation. And here, my question is, how do we use fluorescence quartet to understand local and long-range neuronal circuits involved in behavior paradigms or disease mechanisms? So here we're going to show you a couple of behavior paradigms that we have used internally in mice. These can be used in rodents, rats, as Yasmin mentioned earlier, we can use it in birds. So you can use it in your model of interest. Here what you can see is an elevated T-maze with calcium imaging using the quartet. On your extreme left-hand side is the calcium trace extraction, and right above it is the brain movie from the implanted mouse. So I would like to tell at this point is that not only during imaging sessions as behavior studies, even during surgery, the quartet can be connected during your surgery and you can image it live. You will not be able to see single neuron resolution due to the brain inflammation at that point, but you will definitely see fluorescence. If your viral injection has worked, or if you're in the right region of interest in your transgenic animal, you will see fluorescence, and this will ensure surgical success. Here is another behavior setup. This is an odor behavior memory setting. On your extreme left-hand side is where the animal tries to avoid the extreme corner left corner of this behavior setting. The quartet stays away from the behavior setting and it does not interfere with the animal's naturalistic behavior. On your right-hand side is the bilateral CA1 implantation and it's a brain movie. The field of view is 1 millimeter in diameter. And you can see calcium imaging recording. So during swimming experiments, a lot of you would do swimming experiments, and this is you can comfortably use it with the quartet because the electronics stays away from the head of the animal, and quartet does not get affected with water or if it gets wet. Now, this is an open field setup using the quartet. Quite a number of questions that we get asked is if the fibers are long enough to reach the ends of the open field, the animal can move comfortably, and this is an example of an open field setup. So, as we saw a couple of experimental setups using multiple regions of implantation in a single animal, we move on to, can we use quartet to image multiple mice brains simultaneously in social interaction experiments? Yes, so this is these are bilaterally implanted mice, and this is social interaction studies using the quartet. As you can see, they can comfortably interact with each other. Another way of using the quartet, you don't want to implant multiple implants in the same animal, and you want to have only single implants in different animals. This is a high throughput calcium imaging session using quartet, where 4 unilaterally implanted animals are imaged simultaneously. So it depends on what your imaging session, the length of your imaging session is, and you can use up to 4 animals simultaneously. Another question is, can you combine optical imaging with other modalities like optogenetics, EFIS, MRI? Is it possible? Yes, it is possible because the quartet is small. The lens connectors are pretty small, and here is a combination with EFIS where you can similarly use it with optogenetics. What you can see on the left-hand side is an EFIS equipment, and on the right-hand side is the fluorescence quartet. The mouse is implanted again bilaterally in the hippocampus with 2 lens connectors of the quartet and 2 EFIS probes. here is a zoomed in version of that mouse, and yes, you can see the EFIS probes and the optical imaging probe. This is the animal walking around comfortably during recording of both the neuronal activity with fluorescent proteins and your EFIS recording. So quickly, before I go to the next slide, I'd like to tell that we have data where we have tested quartet in an MRI machine just to get the data if the MRI signals affect quartet signals or if quartet signals affect the MRI signals. And most of any of you who are interested in using an MRI compatible quartet we can discuss it further. So here we talk about what happens if multiple probes are implanted in the same animal. Does it affect the naturalistic or the general behavior of the animal? So here is a control mouse on the upper right-hand corner, and at the lower right-hand corner is a mouse with 3 implants. So the mouse is implanted in the somatosensory cortex in the left CA1 hippocampus and the basolateral amygdala. And if you can see that the animal can comfortably walk around. So these lens connectors are very small. So if there is an important brain region that you would not, if you think will get affected during aspiration, you would not like to touch it, you can implant at an angle. And these lenses are totally customizable. So anywhere, any region of interest, we can customize the lenses, short region, much deeper brain regions like brain stem, and yes, we can customize these lenses for you. Now, another quick poll question. Have you thought of what research questions can be addressed if you can simultaneously perform dual color calcium imaging, GSP-RSP, and dual color optogenetics, channel reduction crimson R, in 4 regions of the brain and spinal cord? So exactly like the quartet, all the 4 regions, but you will have 2 color imaging and 2 color stimulation. Each fiber, if each fiber can give you that capability. Wow. It's quite interesting poll results that we can see here. Seems like a lot of you are interested and you have thought that if there is an instrument that is readily accessible for you to use dual color calcium imaging and dual color optogenetics in the same region, in up to 4 regions, yes, we would like to talk to you. Please do reach out to us in person. We would love to discuss your research, interesting research questions. We looked online at the published results, and unfortunately, we could not find anything in this combination. So we would like to introduce the chromatone to you. What does the chromatone do? Exactly what I mentioned. Each individual fiber will have 3 different illumination colors. Imaging with GSP or GCAM, RSP or RCAM, and optogenetics, which can be channel rhodopsin or crimson R. You can pick and choose any combination that you are interested in, and it can be continuous wave of pulse and can be independently set by the user. The optogenetics protocols are completely customizable, and you can enter your duty cycle, number of pulses, frequency of pulses into the software. The machine can be triggered in and can trigger out with TTL pulses. We had the small video to show what is the capability in each fiber. So there are 3 colors out of the same fiber: blue for imaging GSP or stimulating with channel rhodopsin, green for imaging RSP, red for crimson R. So every combination, blue, blue-green, blue-green-red, blue-red, red-green, yes, comes out of the same fiber. So in this second video, you can see blue for GSP imaging or channel rhodopsin stimulation, red for crimson R optogenetics. In this, you can see green illumination for imaging RSP or RCAM and red for crimson R optogenetics. And here in the 4th video, you can see imaging green for imaging RSP and blue for GSP imaging or channel reduction stimulation. So each fiber will give you all these 3 colors in continuous wave or pulsed mode. And that brings us to the end of our presentation. Thank you for this interactive presentation that we had and for staying with us through the presentation. So I'll hand it back to Liam. And please visit us at SSN 2021. We have a virtual booth, and if you have further discussions about our multi-scope systems, accessories, software, do reach out on the email that you see. Thank you.
Fantastic. Well, thank you, Roshni and Yasaman, for the really fantastic presentation. Great job. So yes, now we're going to move on to the Q&A session. And as a reminder, if you do have a question you'd like to ask, you can submit it now using that Ask a Question panel. All right, first question here today. Do you have to manually define the ROIs from each cell?
I think Yasaman can take that question.
Yeah, sure. Thank you. So yes, you do. Basically, the capability that comes within our software, you will go and manually select the ROIs. Now, there are, of course, a couple of different techniques that are somewhat automated. These are called PCA-ICA, or this is one method. Another method uses non-negative matrix factorization. With those methods, they are basically, these are code where you need to set, first of all, the good parameters for each brain movie. So PCA-ICA has 4 parameters, and non-negative matrix factorization has more than that. And after you find a good set of parameters, and good set of parameters being the parameters where it gives you the least number of false positives while it detects the largest number of neurons. Now, you find these parameters, you run it on your brain movie, and you get a number of segments or potential neurons out. And then you need to go in and manually choose which one of these segments are really neurons and which ones are false positive. So, I mean, I'm calling it semi-automatic because there is a lot of intervention. You need to find the good parameters, and then you need to eliminate false positives one by one. And depending on what type of movie you have in the brain area, so or what type of neurons you have tagged, manual contouring might be the fastest and the most efficient way to go through. But if you have a lot of overlap between the neurons, so say if you have tagged all neurons in hippocampus, then you are going to need to use PCA-ICA or non-negative matrix factorization. Now, one thing that we didn't have time to go through here is, in fact, the workflow surrounding using these methods. And if you would like to discuss further, please do email us, and we'll be happy to meet with you and show you how that is done. Thank you.
All right, fantastic, great answer. We've had a few questions about the use of the multi-scope systems in rats. Is this possible to you?
Oh yes, exactly like how it's done in the mice. And yes, as you saw, it can be used in other species like birds.
So, sorry, can I add something here? So with rats, you can express GCAM depending on the brain area. I mean, researchers who use rats, they say there must be a bit more vessels, so you need to spend more time controlling the bleeding. Depending on the type of the work you do, there might be concern that if the rats will chew through the fibers and so on. So we are actually going to soon start doing some tests with the lab that uses rats, and they are going to basically help us answer this question of how well behaved rats are, if you will, when it comes to these probes. But if you're interested, and if you're interested in testing things out, again, please definitely do reach out to us, and we would love to work with you and answer some of these questions. But in essence, as Roshni said, yes, the technique is used with rats, and you should be able to do it.
All right, fantastic. Thanks so much, Yasaman and Roshni. Next question here: what is the weight of the implant? And maybe I'll expand this question to say, what is the weight of the implant, and does it affect the behavior very much in the mice?
So the weight is less than 400 milligrams, and no, it doesn't affect. As you saw, we have an amount with 3 implants. We have it in birds, and it's very lightweight. It does not affect any kind of behavior.
Fantastic. To a similar question, so we were asked whether you can have different implants in different regions of the brain? I mean, I think you talked about this a little bit, but maybe you could clarify that a bit.
Yes, so we can implant in different brain regions. The lenses are customizable to the region of interest, and you can implant up to 4. So these lenses can even be used in juvenile animals. If we had, as I don't know if we mentioned that. So yes, it is flexible depending on your brain research.
Yeah, here I brought us to this slide because in this mouse with the 3 implants, here you can see the 3 brain regions. So one lens is in the somatosensory cortex, another one is in CA1, and one is in the basolateral amygdala. So yeah, the strength of this technique is that you can go to any brain region of interest, and it can be as deep or shallow as you want. As Roshni just said, one customizes the lenses for that.
Fantastic. And I think that sort of leads into our next question. How deep can you go for mice? Could you do imaging to the PAG or even deeper areas?
So internally, we have tested it in basolateral amygdala and brainstem. So you can go as deep as it has never been shown to interfere any, hinder any movement or any behavior, if that's the question. And we would love, if it's brainstem or any other deep regions, please get in touch with us. And since we've tested it internally, we'll be able to help you troubleshoot the protocol.
Fantastic. Next question here. So thanks for a wonderful presentation. For how long can you use the system to image the same areas in the brains? Is it days, weeks, or how long is it?
Days, weeks, months? Yes, I'll pick all of it. So if you ask me personally, I have imaged up to 6 to 7 months. I don't know if there's a study that requires it. We were just doing internal testing, and yes, 6 to 7 months are comfortably imaged, and it's worked well.
Fantastic, great answer. All right, next one here. So on slide 11, maybe we can navigate back there. On slide 11, there is an image of a mouse with an implant in its spinal cord. Are there any publications showing the use of this platform in the spinal cord?
So spinal cord, yes, I think Yasaman should take that question.
Yeah, so thanks, Roshni. There aren't any publications yet. So this is done with a collaboration, in a collaborating lab. And basically, the implant it uses only one small hole is removed from one vertebra, and the lens and that little connector piece are implanted there, and then the fiber connects. So you don't need to fuse vertebra together for that. And what is imaged, basically what is done for this work until now is to see, because with the spinal cord, unlike the brain, the lens shouldn't touch the tissue. If the lens touches the spinal cord, you are killing the tissue. So if this is the spinal cord, the lens should be sitting on the top. Now, if the lens is not touching the spinal cord and the mouse is moving, the spinal cord is moving. So the question is, can you even image anything when there is so much movement? And so basically what is done is the test where a fluorescence, basically fluorescein, which is a fluorescent tag, it's injected in the blood vessels through the vein. And and this lab, they imaged the spinal vein while the mouse is moving around. And we could see the movement of the vein. And that was actually quite a pleasant surprise, because the movement is only in one direction, so it's not all over the place. And the displacement is actually quite small. And the vein itself, when we are imaging it, it is very, it's not fuzzy, the image. So the movement does not affect the image quality. And the movement is small enough that one can do motion correction on it. So that is the stage that this work is at. We do have actually a whole presentation on the spinal cord. So, again, if this is something you are really interested in, we keep saying reach out to us. So yes, please reach out to us, and we will talk to you and discuss it with you and show you basically what is done so far regarding the spinal cord.
Fantastic. Yeah, great answer. All right, next one here. So are anesthetics necessary? And if so, how might these affect your results?
So anesthetics during surgery, yes, absolutely, they are necessary. And during imaging, no, they are not. The lens connectors are, you can comfortably handle the mouse or the animal without anesthetizing the mouse at any point during your imaging session. We took it into consideration that a lot of learning and memory studies get affected if you use anesthetics. So we teach you how to handle the animal and how to connect and disconnect the probe comfortably.
Fantastic. Yasaman, was there something you wanted to add there?
Oh no, I was just wondering if I should show the movie again where I'm showing the connection and disconnection, but that should be fine. Yeah.
Oh, we have a video on our YouTube channel where we are connecting and disconnecting directly on the mouse. See some videos.
Yeah.
Fantastic. All right. Okay, next question here. So do you have any issues with scarring after surgery obscuring the field of view or changing the focal plane?
So that question, can you repeat that question for me again?
Yes. Do you have any issues with scarring after surgery obscuring the field of view or changing the focal plane?
If your surgery has gone smooth, we have had, for my initial surgical surgery test, I've had like dead neurons and live neurons, and the quartet can't tell you if the neurons are dead or alive because the dead neuron has fluoresces with a different color of green than your live neurons. So when you image it, you can exactly see what you're dealing with. And we have developed a surgical protocol to maximize live neurons and to reduce the number of dead neurons or scar tissue. And even when we change the focus, I have not had a problem.
Fantastic. And I think in the interest of time, maybe we'll just have a few last questions here. Do you have a range of different probe sizes or any plans to introduce different probes for different applications?
Yes, the lens. Yeah, sure. Go ahead. Yeah.
So yeah, I think if by probes it is meant the lenses that get implanted in the brain. In fact, right now there are already multiple lenses and multiple sizes of lenses. So basically, I mean, the work that is being done in different labs is so different from each other and the brain regions. So each lab, they clarify what brain regions they are interested in. And depending on that, the lens is designed to have the good diameter and a good length to be able to image that brain area. In terms of the size of the fiber optics itself, that's a bit of a different story. So these are specialized fibers and they are quite hard to produce. So basically the 1 millimeter diameter is the sensing area and the length of the fiber is 84 centimeters. And that is the standard. And as we showed you, it is pretty much good for all range of behavior experiments. So what matters here is the size of the lens, and we already have the different sizes.
Fantastic. So we've had a few questions about the price. Can you comment on how much is an individual implant in the system?
So the price of the individual implants or the whole system?
So you can address either.
It is I believe, it really depends. So the price of epifluorescence imaging systems is generally anywhere from 60K to 130K. The quartet system is 6 at 80K, and the implants themselves, it really depends on the type of the lens that is required, because it simply depends on the price of the lens, and that really depends on the lens company. So, if you tell us what particular thing you're interested in, we will be able to give you a more precise answer.
Fantastic. And I think we'll make this next question the last one. So in addition to feeding centers in the hippocampus, I'm interested in electrophysiological readings, optogenetics, calcium imaging in pancreatic islets. Is this possible with your system, and how might lenses be inserted or fixed in this place?
We have not tested it in regions other than the brain. So the one thing I would like to suggest is we offer practice lens connectors. If you're interested, you want to know if you can fix it, if it's possible, we can reach the region. I suggest you reach out to us and we can discuss how practice lens connectors, and you can practice your surgeries with it, and that ensures your surgical success. So any other region, please do reach out to us so we can, because this is individual to individual basis. And I quickly saw a question where people have asked if we give support for surgical surgery and neuronal extraction. Yes, that comes with our system. We give all support to make sure that you publish. In short, yes. All the way from your surgical success to your neuronal extraction to synchronizing your behaviors, we help you through everything.
All right, fantastic. Oh, sorry, Yasmin, anything to add?
Sorry, I just I saw a couple of. I know that was going to be the last question because we are out of time. But very quickly, yeah, there are questions about combination with EFIs and optogenetics. And yes, the lens connectors themselves are so small that, say, if you want to do optical imaging in CA1 and you want to simulate, I don't know, the cortex, you can do that. The question is, can you image and stimulate? So you can implant your optogenetic or your EFIs anywhere else that you want in the brain. With electrophysiology, what Roshni showed you, the electrodes are glued to the side of the lens. So in fact, we are collecting the electrophysiology signal from the same area that we are imaging. And that is definitely possible. So that is how the combination with EFIs and optogenetics happen. And if you're working with some areas which are not as common, as Rashi just said, we can provide you practice lenses and, we help you figure it out, basically. Thanks, Mia, for that extra. Letting me do the extra.
I've noticed a lot more questions. Reach out to us, Twitter, LinkedIn, email. Yes, we are available to answer all your questions. I'm so sorry that we're running out of time.
Fantastic. Well, yeah, we have covered a number of really great questions. So thanks so much, not only for the great insights in the Q&A, but also the fantastic presentation. It's really been a pleasure having you two with us.
Thank you very much. Thanks to all our great audience.
Yeah, definitely. Big thanks to everybody for joining us today. And we'd also like to thank Neurescence for sponsoring this event. And so in closing, thanks again for taking part in this Inside Scientific webinar, and we look forward to seeing you again soon. Have a great day, everyone.